Today's topic is nervous system. Introduction. Nervous system controls all activities of our body. It is quicker than other control system. For example, endocrine system. Its functional units are neurons, and about 100 billion neurons are present. There is a interconnection among various parts of nervous system. Now see the structure of neuron. So it is divided into main three part: cell body, this is dendrites, and this is axon. Now cell body contains nucleus and cytoplasm, and in cytoplasm there is a nasal granules. Now process coming out from cell body known as a dendrites, and long process coming out from cell body is known as a axon. Now functions of neuron. Dendrites are basically responsible for receiving information, sensory information from other neurons and sensory receptors. Nucleus provides energy for the neuron to carry out its function. An axon, it carries neurons message to other body areas. Now this axon is covered by myelin sheath, which is basically a covering. It helps speed of neural impulses, and these are terminal branches of axon which form junction with other neurons, which is known as a synapse. Now neuroglia, they are basically a supporting cell in the nervous system. They are found in central nervous system as well as in peripheral nervous system. In central nervous system, there are four type of neuroglial cell: astrocytes, ependymal cell, oligodendrocytes, and microglia. And in peripheral nervous system, there are two type of cell: stellate cells and Schwann cell. Now astrocytes, it maintains blood-brain barrier, provide structural support, regulate ion, nutrient, and dissolved gas concentration, and absorb and recycle neurotransmitters, and form scar tissue after injury. Ependymal cell, it lines ventricles in the brain and center canal in the spinal cord, as well as it assists in producing, circulating, and monitoring CSF, cerebrospinal fluid. Oligodendrocytes myelinate central nervous system axon and provide structural framework. Microglia, it removes cell debris, wastes, and pathogens by phagocytosis mechanism. Peripheral nervous system, stellate cell and Schwann cell. Stellate cell surround neuron cell bodies in ganglia, regulate oxygen, carbon dioxide, nutrient, and neurotransmitter level around neurons in ganglia. Schwann cells surround all axon in peripheral nervous system, responsible for myelination of peripheral axon, participate in a repair process after injury. In short, oligodendrocytes form myelination around axon of central nervous system, and squamous cell provide myelination around peripheral axons. Now, functions of nervous system: sensory function, integrative function, motor function, homeostasis, sympathetic and parasympathetic nervous system. First, sensory function. Now, as we know that there are various type of sensation like touch, pressure, vibration, temperature, stereognosis, proprioceptions. Now, these sensations are carried by sensory nerve fiber, or we can say afferent nerve fiber, towards the brain or to the brain. So, this sensory function is mainly responsible for carrying all the sensation from periphery, from the body to the brain. Integrative function involves interneurons for integration of sensory information. Motor function: muscle contraction and secretion from the gland known as a motor function. So coming from motor area of the cerebral cortex to the spinal cord first, and then to the effector organ like muscle and glands. Along with endocrine, it helps in homeostasis, maintenance of internal environment of cell. And sympathetic and parasympathetic function. Sympathetic division of nervous system prepares the body for emergency condition, and parasympathetic helps to conserve and restore energy. Now, how nervous system is developed? Development of nervous system. Each individual begins as a single cell structure. First fertilized ovum, then there is a series of cell division occurs. Hollow ball of cells are formed, within which a mass of cell forms the embryo proper, in which a neural plate is formed, which modifies to form groove, then grows to form neural tube, which enclosed by the neural crest cell, which form three primary brain structures known as a forebrain, midbrain, and hindbrain. So this is a neural plate. Okay, so it is made up of ectoderm, middle layer is mesoderm, and then endoderm. This is neural plate. So this is embryological structure. At day 19 of embryological life, you can see here there is a formation of groove in the neural plate. Day 21, now this groove becomes deeper. At day 23, you can see here there is a formation of tube. It is enclosed by neural crest cell. Now this neural crest cell gives origin to future dorsal root ganglion. Okay, dorsal root ganglion. Now various vesicles are formed within primary brain structure at the 20 days of embryological life. As we know that primary brain structures are prosencephalon, also known as a forebrain, mesencephalon, known as a midbrain, rhombencephalon, known as a hindbrain, and last is spinal cord. Now primary vesicles are prosencephalon, mesencephalon, rhombencephalon, and spinal cord. Now secondary vesicles are formed at 35 days of embryological life. Now cavities of this prosencephalon forebrain is divided into two cavities. One cavity for telencephalon and another for diencephalon. And from this, in future, there is a formation of cerebral hemisphere from telencephalon, and from diencephalon there is a formation of thalamus, subthalamus, hypothalamus, and neuropituitary. From mesencephalon there is a formation of midbrain. From metencephalon there is a formation of pons and cerebellum. From myelencephalon there is a formation of medulla, and last, formation of spinal cord. So this is primary and secondary vesicles. Now, organization of nervous system. How nervous system is organized? So it is divided into central and peripheral nervous system. Now this central nervous system is made up of brain and spinal cord. Peripheral nervous system made up of somatic nervous system and autonomic nervous system. Now peripheral nervous system, somatic and autonomic. There are two parts of this peripheral nervous system. Now this somatic nervous system basically made up of spinal nerves. 31 pairs of spinal nerves are there. Cranial nerves, there are 12 pairs of cranial nerves. An autonomic nervous system made up of sympathetic and parasympathetic nervous system. Somatic nervous system, as we know that it is made up of spinal and cranial nerves. So first, spinal nerves. Spinal nerves are of 31 pair, which is attached with the spinal cord by dorsal and ventral roots. So 8 pair of cervical nerves are there. They are called cervical nerves because they are originated from the cervical part of spinal cord. Likewise, 12 pair of thoracic nerves, then 5 pairs of lumbar nerves, 5 pairs of sacral nerves, and 1 pairs of coccygeal nerves. So they are originated from different area of the spinal cord. Now cranial nerves, total 12 pairs. So some are afferent sensory, some are efferent motor, and some have both the types, motor and sensory. So they are known as mixed cranial nerves. So total 12 pair of cranial nerves are there. First one is known as olfactory, second one is optic, third one is oculomotor, fourth trochlear, fifth trigeminal, sixth abducens, seventh facial, eighth vestibulocochlear, ninth glossopharyngeal, 10th vagus, 11th spinal accessory, and 12th hypoglossal nerve. Now, principal function of cranial nerves. First and second. First cranial nerve is olfactory and second is optic. These cranial nerves, first and second, are sensory, related to function of olfaction. So first, olfactory cranial nerve is responsible for olfaction, smell, and second is known as a optic for vision. Third, fourth, and sixth cranial nerve are mixed nerve, means they have both function, motor as well as sensory. They are concerned with the movement of eyeball. Fifth is mixed nerve, again concerned with carrying sensation of face and intervening muscle of mastication. So fifth cranial nerve is trigeminal, third is oculomotor, fourth is trochlear, and sixth is abduction. Now seventh cranial nerve, it is again mixed nerve, name is facial, related to taste sensation, supplying muscle of face, nose, palate, lacrimal, and salivary glands. Eighth cranial nerve is known as a vestibulocochlear, which is sensory nerve related to hearing and balance and equilibrium. Ninth is known as a glossopharyngeal. Again, it is mixed nerve involved in taste sensation, and swallowing. 10th, which is known as a vagus, again it is a mixed nerve. It supplies the heart, bronchi. Then 11th is known as a spinal accessory, and 12th hypoglossal nerve. Both this 11th and 12th are motor and going to neck and tongue muscles respectively. So again, this is picture showing various cranial nerve, olfactory for olfaction, then optic for vision, then third, fourth, sixth supplying the muscle of eyeball, then trigeminal facial area supplies the muscle of face, sinuses, teeth, etc. Motor part which supplies the muscle of mastication. Main muscle is masseter for mastication. Then 7th cranial nerve, muscle of face. Then here vestibulocochlear, 8th cranial nerve for hearing. Then glossopharyngeal, 9th cranial nerve. Motor part supplies pharynx, sensory part, tongue, tonsil and pharynx. Vagus, 10th cranial nerve, heart, lungs, bronchi and GI tract. Then 11th spinal accessory, which supplies the muscle of neck like sternocleido, mastoid and trapezius. And 12th cranial nerve is hypoglossal, muscle of tongue. It supplies the muscle of tongue. Now central nervous system. We know that it is made up of brain and spinal cord. Now whole tissue of the brain and spinal cord is made up of neurons and supporting cells, neuroglia. The structures of brain and spinal cord are arranged in two layers, namely the gray matter and white matter. In cerebrum, or we can say in cerebral cortex, gray matter is seen outside and white matter inside. While in spinal cord, gray matter inside and white matter outside. Now, structure of brain. It is the largest and most complex mass of nervous tissue. Weight is about 1.5 kg. It is located within the skull and continuous as a spinal cord from foramen magnum at base of skull. Brain and spinal cord are covered by three layers of meninges called outer dura mater, middle arachnoid and inner pia mater. So there are three layers of meninges which covers brain and spinal cord. Now space. So there are two space, subdural space and subarachnoid space. Subdural space is potential space containing tissue fluid between dura mater and arachnoid mater. First layer and second layer. Subdural means beneath the dura. Subarachnoid space is between arachnoid mater and pia mater, is filled with CSF, cerebrospinal fluid. So this is space between second and third layer, which circulates, CSF circulates through space and is continuous in brain and spinal cord. Now division of brain in three parts. As we know that this is primary brain structure, forebrain, which is known as a prosencephalon, midbrain, mesencephalon and hindbrain, rhomencephalon. Now this forebrain prosencephalon is divided into talon and diencephalon. From talencephalon there is a formation of two cerebral hemisphere, and from diencephalon formation of thalamus, subthalamus, metathalamus and hypothalamus. Midbrain, which is known as a mesencephalon, in which there are two parts, dorsal and ventral part. Hindbrain, which is also known as a rhombencephalon, divided into three parts, pons, medulla oblongata and cerebellum. Now brainstem. Midbrain, pons and medulla together form this structure which is known as a brainstem. So it is a stem-like structure. You can see here midbrain, pons and medulla. Okay. So this is known as a brainstem. And this portion is known as a cerebellum. Cerebellum, which is attached with the brainstem. And this is cerebral cortex. This is cerebral cortex. Now coming to the brainstem, midbrain, pons, medulla. Now importance of the brainstem. All the cranial nerves are situated in the brainstem. For example, from midbrain there is origin of third and fourth cranial nerves. From pons, fifth, sixth, seventh, eighth cranial nerves. And from medulla, 9th, 10th, 11th and 12th cranial nerves. Cerebellum is connected to the brainstem by three peduncles, superior, middle and inferior cerebellar peduncles. And now this brainstem has a important centers for control of respiration, blood pressure and swallowing. So just revise the peduncles. Cerebellar peduncles, superior peduncle, middle peduncle and inferior peduncle, which connect cerebellum to the brainstem. And here you can see in this picture origin of cranial nerves. Okay. So olfactory nerve, from this filament of olfactory nerve, olfactory bulb, then optic nerve. So this is optic chiasma. Okay. From here there is the origin of second cranial nerve. Then you can see here from midbrain there is origin of third, fourth, means oculomotor and trochlear. Then from pons there is origin of fifth, sixth, facial, which is seventh and eighth cranial nerve. And from medulla oblongata, origin of 9th, 10th, 11th and hypoglossal 12th cranial nerve. Okay, so all the cranial nerves are originated from the brainstem. Now telencephalon or cerebrum or cerebral cortex. It consists of two hemispheres. There are two cerebral hemispheres separated from each other in upper part by a median longitudinal fissure, and in lower part connected by bundle of nerve fibers, which is known as a corpus callosum. Now surface of hemisphere is covered by a thin layer of gray matter, which is about 2 to 4 millimeter thick, which is known as a cerebral cortex. So this is structure of cerebral cortex. Now you can see here there is a large convolution. They are known as a sulci and gyri, which is mainly responsible for increasing surface area of the brain. So this elevated portion, it is known as a gyri, and groove-like structure, it is known as a sulci. So sulci and gyri are there, which is mainly responsible for increasing surface area. Now cerebral cortex of each hemisphere is divided into 4 lobes: frontal lobe, parietal lobe, temporal lobe and occipital lobe. And subcortical nuclei lie deep which is known as a basal ganglia. Okay, it is known as a basal ganglia. Now, structure of spinal cord. Gross anatomy. It is a long cylindrical structure 45 to 50 centimeter in length and 2 centimeter in diameter. Lies outside the skull from base of skull. Now, exchange of this spinal cord from foramen magnum to lower border of first lumbar vertebra, and it ends as a cauda equina, horse tail like structure, which is a lumbar and sacral roots. So here you can see exchange of spinal cord, foramen magnum, or we can say upper border of first cervical vertebra to lower border of lumbar vertebra L1. Now, upper end, as we know that it is a continuation of medulla, and lower end called conus medullaris. Okay, so you can see here, this is pointed end, this is conus medullaris, which is continuous with the fibrous cord called filum terminale. Okay, so this is fibrous cord, you can see here in pink color, this is filum terminale. Now, shape is cylindrical with two fusiform shaped enlargement. So one is cervical enlargement, so this is fusiform cervical enlargement, and another is lumbar enlargement, so which gives supplies to upper limbs and lower limbs respectively. Now, meninges, so it is basically covering of spinal cord. So dura mater, it is outermost layer continuous with epinurum of the spinal nose. So this is dura mater, and it is continuous with the epinurum of spinal nose. So this is epinurum of spinal nose, and this is dura mater. Now, arachnoid mater, it is second layer, thin and wispy. Okay, so this one is second layer arachnoid mater, and third layer, which is stick to the spinal cord, is pia mater, bound tightly to the surface of spinal cord. Now, what is the importance of this pia mater? It forms filum terminale, which anchors spinal cord to coccyx, forms denticulate ligament that attach spinal cord to the dura. Now, spaces. Epidural space means external to dura. So now, what is the importance of epidural space? Anesthetic agents are injected here, and it is filled with the fat. Subdural space beneath the dura, which is filled with serous fluid, and subarachnoid space between pia mater and arachnoid mater, which is filled with cerebrospinal fluid. Now, internal structure of spinal cord. So this is basically a cross section of spinal cord. So first of all, from anteriorly, we can see here, it is an anterior median fissure, and this one is posterior median sulcus. And as we know that presentation of gray matter and white matter, in spinal cord, gray matter present inside and white matter outside, and in the brain it is opposite, white matter present inside and gray matter outside in brain, in cerebral cortex we can say. Now, in spinal cord, gray matter present inside, so it is a H-shaped mass. Presentation is H-shaped. Now, gray matter mainly contains cell bodies, neuronal cell bodies, dendrites, and part of axons. And this gray matter divided into horns. So this is anterior horn. It is also known as a ventral horn. This is posterior or dorsal horn, and this one is lateral horn. Now white matter, it is mainly made up of myelinated and unmyelinated nerve fibers divided into 3 columns. It is also known as a funiculi. So ventral column or funiculi, dorsal column or dorsal funiculi, lateral funiculi or lateral column. So gray matter is divided into horns and white matter divided into funiculi or columns. Now centrally there is a central canal, and in front of central canal there is a part of gray matter which connect both the halves of gray matter, which is known as a gray commissure, anterior gray commissure. And this is part of white matter in front of central canal which connect both the halves of white matter, which is known as a white commissure, anterior white commissure. So this is a cross section of the spinal cord. Now gray matter organization. So in gray matter there is a dorsal root which is sensory. It contains ganglion. It is known as a dorsal root ganglion, and ventral part connected with the ventral root, which is motor function, and together it forms spinal nerves. So dorsal root are sensory and ventral roots are motor, which is also known as a Bell-Majendilo. Now subdivision of gray matter of spinal cord into nuclei and into lamina. So there are various nuclei present in gray matter, like posterior marginal nucleus, substantia gelatinosa of Ronaldo, nucleus proprius. Then nucleus, dorsal nucleus, intermediolateral and intermedio-medial nucleus. Then dorsomedial nucleus, ventromedial nucleus, accessory and phrenic nuclei, ventrolateral, dorsolateral and retrodorsolateral nuclei. So there are various group of nuclei present in gray matter. So groups of ventral and dorsal horn neurons. Ventral horn neurons of spinal gray matter mainly involved in motor functions. Functionally, motor neurons are 4 types. A group of ventral horn neurons, dorsal neurons, and neuron in dorsal horn and in lateral horn. Group of ventral horn neurons arranged in 3 mediolateral columns. Medial group, dorsomedial and ventromedial. Lateral group, retrodorsolateral, dorsolateral and ventrolateral nucleus. Central group, phrenic nucleus and accessory nucleus. Groups of dorsal horn neurons. First neuron is substantia gelatinosa of Ronaldo. They are basically small cells made up of gelatinous material. That's why it is called substantia gelatinosa. Traversed by fibers of dorsal nerve roots which convey pain and thermal sensation, and it has role in gate control of pain. So it is one of pain suppression system. Second neuron is nucleus proprius, composed of internuncial cell and tract cell. Dorsal nucleus composed of tract cell, which receive proprioceptive touch and pressure sensation from trunk and lower limbs. And it is also called thoracic nucleus or Clark's column, and it extends from C8 to L2 segment of spinal cord. And fourth is posteromarginal nucleus. Now neurons in dorsal horn, which is mainly involved in sensory function. Dorsal horn neuron are of two types, internuncial and traxal. Internuncial neuron located between sensory fibers terminating in the dorsal horn and motor neurons originating in the ventral horn. That's why it is called internuncial neuron. The traxal, these cells receive impulses from various receptors of the body through dorsal nerve root fibers. Axon of this cell enter the white matter of the spinal cord on the same or opposite side and constitute either intersegmental tracts or ascending tracts which terminate in various masses of gray matter in the brain. Neurons in lateral horn. It is also known as a neurons of intermediolateral group of visceral afferent neurons, which exchange from neurons present in T1 to L2 lateral horn. So T1, that is thoracic segment, L is lumbar segment. These are preganglionic neuron of sympathetic nervous system. Their axon terminate in relation to postganglionic neuron in sympathetic ganglia. Neurons present in S2 and S4 lateral horn, sacral root. These are preganglionic neuron of sacral component of parasympathetic nervous system, and their axon leave spinal cord through ventral roots to reach spinal nerves, for example pelvic, splanchnic nerves for pelvis and abdomen. Now division of spinal gray matter into lamina. Now this lamina is known as a Rexad lamina, divided into 10 lamina. So lamina 1, it is basically correspond to posterior marginal nucleus. So this is lamina 1, it is correspond to posterior marginal nucleus. Lamina 2 correspond with the substantia gelatinosa of Rolando. Lamina 3 and 4, it is correspond with nucleus proprius. Lamina 5, neck of dorsal gray column. Lamina 6, dorsal nucleus in base of dorsal gray column. Lamina 7, confined to lateral gray column. Lamina 8 and 9, confined to ventral gray horn. Lamina 10 forms gray matter around the central canal and consists mostly of neuroglial cell. White matter, as we know that it is formed by nerve fibers arranged as ascending and descending tracts, and it is divided into right and left halves. Each half exhibits following parts: posterior funiculus, anterior funiculus, and lateral funiculus. Now presentation of white matter. So most of ascending and descending tract passes through this white matter. So there are a list of ascending tracts like dorsal column tracts, spinocerebellar, then lateral spinothalamic tract and anterior spinothalamic tract. And in descending tracts there is a lateral reticulospinal, lateral corticospinal, rubrospinal, medial reticulospinal, anterior corticospinal, vestibulospinal and tectospinal tracts. So these are extrapyramidal tracts, descending tracts, motor tracts. Spinal segments consist of 31 spinal segments. 8th cervical segment gives origin to 8 cervical nerves. Likewise, 12 thoracic segment, 12 thoracic nerves, 5 lumbar nerves, 5 sacral nerves, and 1 coccygeal segment gives origin to 1 coccygeal nerve. So each spinal nerve is mixed nerve formed by union of two roots, dorsal and ventral. Dorsal root is sensory and ventral is motor. Now dorsal nerve root, it is formed by several rootlets attached to the surface of spinal cord. All sensory fiber reach spinal cord through dorsal nerve roots. Each dorsal nerve root is marked by swelling known as a dorsal nerve root ganglion or spinal ganglion, which is composed of T-shaped unipolar neuron with peripheral and center process. Now this peripheral process, it exchange up to the sensory receptor in the skin. That's why area of skin supplied by spinal nerve is known as a dermatome. And center process has two division, medial and lateral division. Now medial division is myelinated group 1 and 2 fibers, which is mainly responsible for carrying proprioception, then touch, pressure and vibration. So group 1 mostly carries proprioception and group 2 touch, pressure and vibration. Lateral division in third group and fourth group. Now third group is thinly myelinated group and fourth group is unmyelinated fibers. Fast and discriminative pain and temperature sensation carried by group 3, slow pain and visceral sensation carried by group 4. Now last is function of spinal cord. We will discuss in next lecture. Thank you.